start it again. So before I start talking about transplantation immunology, is there anybody during the break thought, oh, I should have asked him about this in respect of tumour immunology? Has anybody thought of any questions they'd like to ask related to the stuff I talked about before the break? No? Sure? Okay, well, you can always come up to me at the end, after I finish today. Anybody that wants to come up at the end and ask me questions, chat, that's absolutely fine. Feel really comfortable to do that. So, now we're going to talk about another situation. We talked about how tumours maybe can be recognised by the immune system. We heard that the immune system has evolved to fight infection. So over a huge period of time during evolution, the immune system has developed many different types of cells and molecules to fight infectious agents. But also it seems that it can fight tumours. What it recognises, as we heard, in general, is things that are non-self. So it becomes tolerant of self antigens, but it responds to non-self antigens. And I mentioned briefly during the first session that one of the things that it can recognise is a transplant. I talked about people receiving transplants being immunosuppressed. Do they develop more tumours? And the answer is yes. Their immune system is dampened down, and people that are immunosuppressed because they're receiving a transplant do develop more tumours of certain types. But what about in the transplant situation itself? There we have a foreign organ or tissue being transferred from one individual to another. It's foreign, it's non-self. So how does the immune response see those kind of transplantation antigens, and what can we do to stop the body rejecting transplant? That's what I'm going to talk about. Now it's important to start off thinking about the different situations in which you can transplant an organ or a tissue. So you can have this was what this you can have what is called an autograft. And an autograft is where you take something from one part of the body and transfer it to another part on the same individual. And this is very often done when somebody has burns. So if you have a burn here, or sort of here, then maybe you take some skin from here and transplant it to an area that's visible. So that's a very common situation. And there you're taking the tissue from one part of the body, and in the same individual, put it somewhere else. So it's not foreign. It's that part of that person's own body tissues. So in that situation, it's an autograft, and usually that doesn't require any immunosuppression. Or you can have an isograft, where between genetically identical individuals, for example, monozygotic twins, or an inbred strain of mice. So if you take mice and you brother systemate them over 20 generations, so you keep taking the mice and brother systemating the mice, then you end up with an inbred strain of mice, where the genes are all identical in each mouse. And in that situation, in an inbred strain of mice, you can transplant skin from one CBA mouse, for example, a strain CBA mouse. You can transplant from one to another CBA mouse, and the graft is not rejected. It's not seen as foreign because the genes in those two mice are absolutely identical. Now, in a human, the only time you see that situation is in genetically identical individuals who are monozygotic twins, derived from the same zygote, and therefore their genes are identical. But most people don't have an identical twin. So most situations, that's not going to be something that you can use in a transplant. Occasionally it will be, but it's very rare. So in most situations, in the human, what you have is what is described as an allograft. And an allograft is where you have a graft between different members of the same species. Okay. So Mr. Smith to Mr. Jones, If you're in England, I don't know, Mr. Wang to Mr. Zhao or whatever, if you're in China. So different individuals, but both human, okay? And there, the genes would be different, so that the graft would be seen as foreign by the recipient, and the recipient's immune system would recognise the graft as foreign and react against it, just as it would react against a virus or bacteria. And then increasingly, people are beginning to talk about xenografts. And this is where you have a graft between members of different species. For example, monkey to man. Now, it's unlikely for a variety of reasons that organs would be transplanted from monkeys to men. But people are beginning to think in a very serious way about transplanting organs, for example, from pigs to humans. I'll talk about that at the end of the session. Now, there are various rules that govern transplantation, and these rules were worked out by a number of people, including somebody called Sir Peter Medawar in London. And really, the impetus for this work came from the Second World War, where, again, people were getting burns. We heard about people getting burns, and in this case, they were wanting to try and do transplants, not in the same individual, but between individuals. And he was looking at mice to try and work out what rules there were that governed the transfer of organs and tissues between individuals. And this is what he found. If you take mice from strain A, and then if you take mice of strain A, you take a skin graft from mice of strain A, and transfer it to another strain A mouse. So an inbred mouse, genetically identical, then that skin graft is accepted. However, if you take a skin graft from mice of strain B and transfer that to mice of strain A, the genes are different, it's a foreign graft, and it's rejected. If you take strain B again, take a graft from there, but now you put it into what we call an F1 hybrid, the offspring of mating strain A mouse and strain B mouse, A times B mouse. If you take a skin graft from a B strain mouse, put it into an F1 A times B, it's accepted, because this mouse has a mixture of genes from strain A and from strain B. So strain A is self to it, and strain B is self to it. So it doesn't react B because it's self-antigen. Okay. But if you do this the other way around, you take... I don't know how that works. I'll learn. If you do it the other way around, take an A times B F1 and take skin from this and put it into a B strain mouse, then it's rejected, it's not accepted. Because half of this mouse, half the genes in this mouse are strain A, and strain A is foreign to a B strain mouse, so it gets rejected. And these were what you could call the rules of transplantation. And this was done in mice and became the foundations for how we view transplantation in humans in terms of trying to make the best match possible to try and get a graft to be accepted. Now, just like an immune response to an infection, so an immune response to a graft can show immunological memory. The basis of vaccinating somebody against infectious agents is to induce immunological memory. This is a property of the adaptive immune response. The only cell type in the body that shows immunological memory is the lymphocyte. No other cell type in the body shows that. Lymphocytes are part of the adaptive immune response. And immunological memory is produced in response to infection, but it's also produced in response to grafts. So if we take a mouse that is an A strain mouse here, and we put a B strain graft onto it, and then we take T cells from that mouse. So a mouse that's A strain and has been grafted with a B strain skin graft, we then take T cells from that mouse and transfer those T cells to a naive mouse. In other words, an A strain mouse that hasn't seen a graft before. Okay. And then we graft B strain skin onto this A strain mouse. Okay. So now This mouse has never seen that graft before, but it has received T cells from a mouse that saw that graft. And you get a type of rejection that is referred to as second set rejection. And this is essentially the same as a secondary immune response. It's a faster response. And in terms of transplantation immunology, people talk about a second set response. But it's basically a secondary response, and it's a fast rejection. Okay. However, if you do the same experiment, you take an A-strain mouse, you graft it with skin from a B-strain mouse, you then take T cells from this mouse and transfer it into a naive A-strain mouse. But then instead of giving this mouse the graft from a B-strain mouse, you give it a graft from a C-strain mouse. So not only has this mouse never seen skin from a C-strain mouse before, but the T cells that it's received has never seen the antigens associated with the C skin. So then you get essentially a primary response, or in other words, a first set rejection. And if you do this the other way around, so you take A and you do a C skin graft, and then you transfer the cells, and you then do a B graft, again it's a first set rejection, because it hasn't seen the antigens from that mouse before. So just like an immune response to an infectious agent, the immune response to a transplanted organ or tissue shows immunological memory. And it's specific. It's specific for the particular antigens. It recognizes A-strain skin, it's specific to those antigens. And this is just a time course of kind of rejection you get. So in an autograft you don't get any rejection, but in first set type of rejection where there's no immunological memory, then after a certain amount of time, 16 days or so, 50% of the grafts have been rejected, 50% survive. Whereas in the second set rejection, where they've already seen the antigens, they're primed to these antigens, there's immunological memory, then the rejection is much faster. And about half the grafts are lost after about 8 days. We know that the immune system is involved in rejecting grafts because you produce antibodies against the foreign HLA type in the graft, but also, this is a kidney graft, you can see lots of lymphocytes infiltrating the graft. So there's an immunological rejection, and it's mediated eventually by an infiltration of immune cells into the graft. So how is the graft recognized? Well, as I said, you get antibodies, but you also get T cells being activated. And there are essentially 3 different pathways by which T cells can be activated to recognize the antigens in a foreign graft. This is a little bit complicated. I'll go through it reasonably slowly. So when you transplant an organ, we can talk about the donor, the person giving the organ. So let's say it's a kidney. Okay? So you have a kidney donor, and then you have a recipient. Okay? When you give a kidney, or almost any other tissue, there are kidney cells there, but there will also be some immune system cells there. It's hard to wash out all of the immune system cells out of a graft. So there will always be some donor antigen-presenting cells there. Okay? So dendritic cells, macrophages, B cells, other cells that can act as antigen-presenting cells. You can't wash them all out of the graft. You can't get rid of them all. So when you transplant a graft, there will be some donor antigen-presenting cells there. Obviously, the person receiving the graft has an immune system, so they will have all the cells of their immune system, including T cells, just like we all have T cells. So when you transplant a graft, there will be some donor antigen-presenting cells present in the graft that is received, and the recipient will contain T cells. The donor antigen-presenting cells will have on their surface MHC molecules, and if they're professional antigen-presenting cells, it will be both class 1 and class 2. And these MHC molecules obviously potentially can present peptides. So that's what MHC molecules do. They present bits of protein actin that have been chopped up by proteases into short peptides. So these MHC molecules can be presenting peptides. This MHC is on the donor antigen-presenting cell. So it is foreign to the recipient. And we refer to MHC that is different to that in the recipient as allo-MHC. Okay? So this is allogeneic, genetically different MHC, because it's present on the donor antigen-presenting cell. It's being recognised by the recipient. And this allogeneic MHC can present peptides, because all MHCs are different, and we'll talk about this in a second. Everybody's MHCs are different to each other's. There may be some that are the same, but others will be different. So collectively, your MHC is different, and that's why, fundamentally, that's why you reject a graft. But because this MHC is different to the MHC in the recipient, then it can be presenting peptides that the recipient will not be tolerant to. The recipient will be tolerant to peptides produced by their own MHC molecules. But they won't be tolerant to peptides presented by this allo-MHC, because that MHC is different and therefore will present different peptides. Okay? So you get allo-MHC with multiple non-tolerant, they're not tolerant, self-peptides. They're self to the donor APC, but they're not self to the recipient in their form of being presented by the MHC. Or you can get what's called the semi-direct pathway, where you have allo-MHC that is on the donor cell. It's just like this, but it might be on an antigen-presenting cell, it might be on any other kidney cell, for example. And this MHC then gets taken off by a recipient antigen-presenting cell. So now this antigen-presenting cell is in the recipient, not in the donor. So it's different to this situation. And it's thought in some cases, and it's not clear how often this happens, that this combination of peptide plus MHC is transferred from the donor cell, so this could be a mesangial cell in the kidney or any other cell in the kidney, could be taken off by the recipient APC intact. So it's not degraded, it's not changed, just the MHC with the peptide in it is taken off by the recipient APC and then shown to T cells in the recipient. This is referred to as the semi-direct pathway. And again, this should be recognised because it's from an MHC. Or a third situation, which is referred to as the indirect pathway of antigen recognition, is whereby you have allo-MHC on the donor cell. So again, this could be any cell from the kidney at all. It doesn't need to be an antigen-presenting cell. That MHC is taken off by the recipient antigen-presenting cell, and then just like proteins normally, what happens normally to proteins in an antigen-presenting cell, they get processed and then presented. And processing means they get chopped up into peptides. And you can chop up MHC molecules just like you can chop up any other protein. So peptides that are part of the polypeptide sequence of the MHC, if you have MHC class 1, the heavy chain is 43 kilodaltons, for example. It's a large protein. That can get chopped up by proteases into short peptides. And those peptides that are derived from the allo-MHC can then be put together with the recipient's own MHC. So you see I've coloured this green rather than brown. Okay? So this is not donor MHC, this is recipient MHC. Okay? But it's presenting a peptide, a brown peptide here, that is derived by chopping off the MHC. And because the MHC is different in the donor from the recipient, so the peptide would be different. It wouldn't have seen that peptide before. So it's using its own MHC. So it's self-MHC, but it's presenting allopeptide. Peptide is allogeneic because it's derived from a different MHC molecule to the one of the recipient. So those are three different ways in which T cells can be stimulated to recognise antigens derived from the foreign graft. Now there's another situation that happens which is a rather strange situation, a rather odd situation. Because normally when you transplant an organ or a tissue from one individual to another, the problem is that the recipient recognises the graft and rejects it. If you give somebody a kidney and you haven't tissue typed it, you haven't matched the donor and the recipient, and you don't give immunosuppressive drugs, then the recipient rejects the kidney because it's foreign. But I've already mentioned that in a tissue, you have some immune system cells. If you're giving a kidney, you can't get rid of all of the immune system cells. And sometimes the opposite happens. The graft rejects the recipient. So the immune system cells in the graft actually starts attacking the recipient. It's very odd, but it does happen. And there's one situation that it happens a lot and is a very, very big problem in clinical medicine. And that is in bone marrow transplantation, or nowadays often hematopoietic stem cells that are derived from peripheral blood. Because these are cells of the immune system. You're transferring, in a kidney, you're transferring a kidney and there may be a few immune system cells there that you haven't managed to get rid of. In a bone marrow, you're transplanting just immune system cells, because the bone marrow is where you make your immune system cells. So then you're transferring a lot of cells, and you have to immunosuppress the recipient to stop them rejecting the graft, but you're giving them lots of immune system cells, and the graft can then actually very often reject the recipient. It's called graft versus host disease. It's a major, major problem in bone marrow transplantation. And this is what happens, this is shown in a mouse, that here you have a mouse that is a B strain mouse. It's mating of a bee with another B strain mouse, so it's a pure inbred strain bee. It's labeled BB here. If you give this to an animal that is A times B, then B is not foreign to this mouse, so it doesn't reject it. But because this mouse, as well as having B antigens, also has A antigens, these immune system cells will recognize the A as foreign and will attack the recipient mouse, and you'll get graft versus host disease. And the same happens if you transfer to an A times A mouse that you've irradiated. Now, if you didn't irradiate this mouse, because this is completely different, this is BB, this is AA, this mouse would reject this. But if you irradiate this mouse, then you damage this mouse's immune system. So it's not able to fight the graft, but the graft can attack this, and again you get graft versus host disease in that situation. And this is what it looks like in a patient. It's very, very unpleasant, and often patients will die from graft versus host disease unless one does something to stop them dying. Now, I would have liked to have shown you a slide of the number of transplants performed worldwide. I was not able to find such a slide, so I'm afraid this is biased in that it shows figures for the United States of America. But at least it gives you some idea of the numbers of different types of transplants that are done, and what types of organ are transplanted a lot, and what types of organ are not transplanted so much. And of course, the organ or tissue that is transplanted by far the most is blood. Blood transfusion. That is a transplant. You're transplanting blood from one individual to another. And that is very, very common. It's been done for many, many decades and is very successful. I mean, that is saved millions and millions and millions and millions of people's lives, transplanting blood. You need to match. You need to match for the major antigens, ABO antigens. But there are many, many other blood group antigens. There are actually hundreds and hundreds and hundreds of blood group antigens. You said the major ones that you need to match for. The ABO is the one you really need to match for, and also sometimes rhesus antigens. But the other ones you don't need to match for. And in the USA, for example, it's estimated that over 40 million units of blood are used each year. And it's a very, very successful technique. But what about other transplants? Well, kidney is one that is very successful, and these figures go back a bit in time, but in the year 2000, over 13,000 transplants, kidney transplants in the US. Bone marrow transplantation, we talked a little bit about, rejection rare, but graft versus host disease is a big list, that's what I was telling you. Liver, quite common, nearly 5,000 transplants in the USA in the year 2000. Heart, also quite common, 2,000 per year and so forth. Lung, cornea is an interesting thing because the cornea doesn't have a blood supply. So you can graft corneas without any immunosuppression. However, you can get immunological rejection of cornea. People think that cornea, you don't need to think at all about immunological rejection, but sometimes you can get immunological rejection. Sometimes corneas can become vascular. But in general, they're not, and therefore immunosuppression usually is not required. And a very, very common procedure, 40,000 transplants per year in the USA. So you can see lots of different organs and tissues now can be transplanted. And this actually gives some numbers, and the reason I've shown this in addition to the previous slide is really to show you how over the last decade the numbers have changed. And you see that they've gone up a bit, but not hugely. The numbers have kind of stabilised. So we've reached a situation now where the logistics of doing transplantation have all been worked out. We know about how to match the donor and recipient. We know what antigens you need to match, which are mainly the MHC antigens. We have developed good immunosuppressive drugs that we can utilise. And the problem now, the reason that this is kind of levelled off, is that there aren't enough donors. So there are plenty of people needing kidneys, lungs, hearts and so forth, but there are not enough donors to provide the need. So the graph has plateaued, not because of any problems with the technology, but the fact that there aren't enough donors. And I'll come back to that at the end of the talk. Even if you do a good match, even if you immunosuppress, there is no guarantee that a graft will survive. And I think there's an important difference to appreciate in two different terms that are used. That is graft survival and patient survival. They're not the same thing. Okay? You can have a kidney graft, and maybe after 5 years you reject the kidney graft, but you don't die. So you survive, but the graft dies. Okay? So the graft survival and patient survival are two different things. Okay? The graft won't live longer than the patient, but the patient may well live longer than the graft. And you can have another graft. You know, you reject a kidney, maybe you can have another one. And this shows the survival rate of grafts, another patient with grafts, at 1 year, 3 years, and 5 years for various types of grafts. And you can see that it's a pretty successful technique. If we look just at 5 years survival, okay, for heart, 70% of patients, 5 years after their transplant, the transplant is still working absolutely fine. Okay? So others are not so successful. If you try and do heart and lung together, it's not as successful as heart on its own. Pancreas, after 5 years, only about 50% are surviving. The graft, only about 50% of the grafts are surviving. But it's a pretty successful technique. So when you do get rejection, what is it that causes the rejection? Well, there's still a lot that is not known about the mechanisms behind the immunological rejection of foreign grafts. There's still a lot that isn't that well understood. But what is known is that sometimes you can get an almost immediate rejection. And this is due to preformed antibodies. So you get what's called a hyperacute rejection. This happens often within a few minutes, certainly within hours. And this is because the recipient already has antibodies that are present that recognize antigens in the donor, and those antibodies bind to the transplanted tissue, complement becomes activated, and it's rejected very rapidly. That is something that it still does happen sometimes, but it really shouldn't happen these days, because we know the antigens that one needs to match, and we can test whether the patient has antibodies against those particular antigens. So it's something that isn't seen too often, but it does occasionally. Then you can get what's called an accelerated rejection that happens in days rather than minutes or hours. And this is where there are T cells present that become reactivated. And these T cells have already been sensitized to those foreign antigens. So they're memory cells that go straight into making a secondary immune response. What is much more common is acute rejection that happens after days or weeks, and this is due to T cells recognizing antigens, mounting a primary immune response, so they're recognizing foreign antigens, They haven't seen those antigens before, so there's no memory cells. They have to generate a new primary immune response. It takes a while to get going, and it takes weeks usually before the graft starts being rejected by this acute rejection. Those three are pretty well understood in terms of the immunology. What is much less understood is why some people reject grafts after 5 years or 10 years. What is it that, if you don't recognise it as foreign, either making a primary immune response or you already have some sensitisation, you make a secondary immune response. Either way, you're going to reject it within days or weeks. Okay? But why would you reject it, after 5 years or 10 years? And that really isn't well understood. But it's thought that maybe you begin to produce antibodies for some reason you didn't, but now you start making antibodies, that you're producing immune complexes that maybe get trapped, that you maybe have some kind of slow cellular reaction going on. Or often it might be that you have a recurrence of the disease. This is nothing to do with immunology. If you had a kidney transplanted, you had a kidney transplanted because you had kidney failure. What caused the kidney failure? That wasn't anything to do with the immune system. There was something wrong that caused the kidney failure. And maybe whatever it is was causing your kidney failure, that occurs again with a transplanted kidney. Whatever metabolic process was going on in the body that damaged your original kidney, maybe the same thing ultimately will happen to your transplanted kidney. So that may well be, in most cases, the reason for chronic rejection that occurs over a period of months or years. So from those experiments looking at the transplantation of skin in mice, it was worked out that the genes that are really important, that are different from one individual to another, and that are really important in terms of rejecting a tissue or organ, are the genes of the MHC, the major histocompatibility complex. In mice that's called H2, but in human it's called HLA. It has this name, it's a rather unfortunate name, because these molecules were first discovered on leukocytes, white blood cells. So those molecules, which can be recognized as foreign in another individual, therefore they were described as being antigens, because the immune system recognizes antigens, that are found in the humans. So they're called human leukocyte antigens. But it was a bit too rapid to give them a name, because then people started looking and found, they're not only present on white blood cells, leukocytes, they're also present on every nucleated cell in the body. So leukocyte is not a good word to use there. But that's what stuck, and they're called HLA. So these are molecules that are present on the surface of all of our nucleated cells. Now, they did not evolve to stop surgeons transplanting organs and tissues. That's not why we have MHC molecules. We have them to present bits of peptide from viruses so that cytotoxic T cells can recognize something as being foreign, or being presented by MHC class 2 on professional antigen presenting cells so that CD4 positive helper T cells can recognize something as being foreign. That's why we have MHC. It's just an unfortunate byproduct that we get this rejection. Now you could ask, well, why do we need to have different MHC molecules? And that is related to the fact that infectious agents, particularly things like viruses and bacteria, are changing all the time. They're mutating all the time. Many bacteria divide every 20 or 30 minutes. Every time nucleic acid is replicated, you can incorporate new nucleotides into the nucleic acid. So you can make mutations very rapidly. And infectious agents are mutating, they're changing all the time. You'll all be familiar with, influenza virus. You need a new influenza jab every year because the influenza virus changes every year. And MHC is thought to have evolved so that at a population level, if we have lots of diverse MHCs, then whatever mutations a virus carries out, some of us will have an MHC which can bind a peptide from that virus. Because what MHC does is bind peptides. And the difference in MHCs between you and me and you are in the MHC binding groove, the bit of the MHC that binds the peptide. That's where most of the amino acid differences are between one individual's MHC and another individual's MHC. It's thought that evolution has generated this huge diversity. We call the MHC the most polymorphic genes in the human genome. It means the most diverse genes in the human genome. And that is almost certainly to fight infection population level, some of us will survive however many mutations the virus makes. Okay? But it does mean that once transplantation was developed, which after all is a very recent, it's only 50 years or so that transplants have been carried out in humans, it means that we have to tissue type. We have to match the MHC of the donor with the MHC of the recipient. Now this is a diagram illustrating the MHC genes, and although I made this 2 or 3 years ago now, it's still pretty much common, pretty much coloured. Some of these numbers might be very slightly higher, but what is important is just to look, first of all, at the fact that at the top you see something labelled M, and at the bottom you see something labelled P. This is to indicate that the MHC is what is described as co-dominantly expressed. That means that you've inherited some MHC genes from your mom, and you've inherited some from your dad, and you express both of them. Okay? So this is the maternal chromosome, or the one you've inherited from your mom. This is the paternal chromosome. The MHC in human, the HLA in human, is on chromosome 6. Okay? So this would be chromosome 6 from your mother, chromosome 6 from your father. And they have one part of chromosome 6, you have the genes of the HLA. And there are quite a few of them, but there are 6 that are particularly important. Okay? And these are the 3 main MHC class 1 genes and the 3 main MHC class 2 genes. The MHC class 1 genes in the human are called HLA-A, HLA-B, and HLA-C. And it's not because I don't know my alphabet that it should go A, B, C. It's because that's the way they're organised on the chromosome. Actually, in that order on the chromosome. And then the 3 major MHC class 2 genes, there are some minor ones that are important in some situations. We maybe talk about that at the very end of the talk. The 3 main ones that are important from the transplantation point of view are HLA-DP, DQ, and DR. Now these genes, the actual sequence, the nucleotide sequence of these genes varies enormously from one individual to another. So the sequence of my HLA-DP will probably be different to yours, and yours will probably be different to yours, and yours will probably be different to yours. Okay? Some of them are quite common. Some variants, some versions of MHC are quite common. Okay? But they do, in general, vary from one individual to another. So perhaps you have inherited from your father on the paternal chromosome the version of HLA-DP that we call DP6. But from your mother, you've inherited DP2. And therefore, if you look at the colours here, red and a pinkish colour, at the top there, top left, this individual is expressing the version of HLA-DP they've inherited from their mother, which is HLA-DP2, and the version of HLA-DP they've inherited from their father, which is HLA-DP6. And the same for each of these. So at the top we have the 3 class 2 genes, and maybe you have a different version from each parent, so you could have 6 different MHC class 2s being expressed. At the bottom we have the MHC class 1s, and again, maybe you've inherited 1 from HLA-B, B8 from your mother, B27 from your father, for example. Okay, same argument applies. So you could have 12 different versions, okay, of MHC on the surface of your cells. Now this would have to be a professional antigen presenting cell, because professional antigen presenting cells, as well as expressing MHC class 1, which is down here, also express MHC class 2. Whereas most cells on the body, in the body, only express MHC class 1. But still you'd have 6 different versions. Okay? Now the numbers that I put here are the numbers of different versions that have been described. So for HLA-BP, for example, it consists of an alpha chain and a beta chain. There are 23 different versions of the alpha chain and 121 different versions of the beta chain. And many different alpha chains compare with many different beta chains. So the variety is huge. Okay? So that's why, unless you have an identical twin, then a graft would be rejected unless you match for these various antigens. So you have to do tissue typing, you have to find out which HLA versions the donor has which HLA versions the recipient has and make sure that they match. So we can then ask the question, well, you've got 3 different MHC class 1s, A, B and C, 3 different MHC class 2s, B, P, D, Q, D, P, D, Q, D, R. Which is the most, are they all equally important or are some more important than others? Okay. And this is going some way to addressing that. So if you take, this is looking at present class survival after 1 year, this column. And if you take a situation where there are no mismatches for HLA B and C, okay? So HLA A is identical, no mismatches for A and B. So HLA A is identical between the 2 individuals, HLA B is identical between the 2 individuals. If there are mismatches at HLA-DR, the class 2, what is the consequence of that? So if there are no mismatches, then the class survival at 1 year is 86%. But even if both HLA A and B are matched, so there are no mismatches at A and B, if there is one mismatch at DR, this goes down from 86 to 83. So it's not a huge difference, but it is a significant difference. It's actually statistically significant. If there are 2 mismatches at DR, then 80% survival at 1 year. So again, a bit less. We ask it the other way around. What about if you don't have any HLA-DR mismatches, but you have mismatches at either HLA-A or HLA-B? Well, there you see that if there are no mismatches, at 1 year there's 88% survival. With no mismatches, 1 mismatch, 84. 2 mismatches, same kind of value. 3, probably no difference. 4, a bit of difference. So what this is telling you is that matching is important, but it's not actually hugely important. It is important, but you can live with some mismatches and still get reasonable survival if you give immunosuppression as well. All these patients are also receiving immunosuppression. So a better match is better, but the degree to which you need an absolute match is probably not that critical. And this gets debated in the literature. People do debate a lot about whether particular HLA types are important to match. Some people think that you can completely forget about HLA-C, for example, that you only need to think about matching HLA-A and B, the class 1, and that you can just forget about matching HLA-C. Other people think that it is important to match HLA-C. So there's still a lot of debate that goes on in the literature. But you can see that it makes a difference, but as long as you've got some matching, then you can probably not worry too much about some mismatching. But in all of these situations, you absolutely have to have immunosuppression, because without immunosuppression, for sure, the graft will be rejected. And there are new immunosuppressive drugs being developed all the time. Better ones are being produced, but we've probably reached a stage where we've got, in terms of immunosuppressive agents, we've probably got most of them that we would kind of need. But I'll expand on that again in a second. But if we look at survival here, so this is survival with 2 immunosuppressive agents that were very commonly used as a combination until relatively recent. Azathioprine and corticosteroids, which are immunosuppressive agents. If we look at survival after a number of months, after 1 month, 60% survival with this combination. After 3 months, 40% survival. This is liver transplant. After 6 months, 35% survival. So these are old drugs that used to be used. So they were okay, but they weren't great. And then a new drug was discovered about 20 years ago called cyclosporine. And this has become one of the major drugs that's used to immunosuppress recipients that are receiving transplants. Used very, very widely. And you can see that once cyclosporine was introduced, and that it replaced azathioprine, so instead of using azathioprine plus corticosteroids, use cyclosporine corticosteroids, which is in the blue. You see it made a huge difference, especially when you go out into years rather than months. Huge difference. 60% survival of the transplants. This is graft survival rather than patient survival, compared with 25%. So this was a big, big leap forward to the development of this drug called cyclosporine. And now we have lots of different types of immunosuppressive drugs. And I'm not going to go through this list in any great detail. The corticosteroids I've already mentioned. Cyclosporine is an example of a calcineurin inhibitor, and I'll show you a slide in a second showing that. But there are other drugs that are very similar, or have similar type of action. One called tacrolimus. And cyclosporine is the most commonly used drug. There's a bit missing off this slide. It's the most commonly used drug these days in heart and lung transplants. It's usually given with other drugs such as azathioprine, as we just said. And then tacrolimus is very commonly used in kidney, liver, pancreas, and small bowel transplants. Then we have drugs such as azathioprine, which is a purely metabolism inhibitor. There's a drug called mycophenolate moscatel, rapamycin, which I'll show you in a second. And then you can use antibodies that suppress the immune system. And these have been known about for a long time. You can use antibodies that bind to lymphocytes and stop the activity of lymphocytes. And if you have polyclonal antibodies, it's referred to as anti-lymphocyte globulin, or you can develop lymphocyte-specific antibodies specific against T cells. And these, if you develop them against T cells that are developed in the thymus, you have anti-thymocyte globulin, or ATG. And these can work very effectively. But these are polyclonal antibodies. Monoclonal antibodies are much more specific. So people are using monoclonal antibodies, such as one that's called OPT83, which binds to a molecule called CD3. And CD3 is present on the surface of all T cells. If you're a T cell, you're going to have CD3 on the surface. If you ain't a T cell, you won't have CD3 on the surface. As simple as that. Because it's a molecule that's required to transmit a signal into the cell when T cell receptor recognizes that. So you can dampen down the immune response of T cells using anti-CD3. You can stop the activation of T cells via antibodies to the interleukin-2 receptor, because interleukin-2 is required for T cell growth and activation. And then people are beginning to develop some new therapies that are maybe more specific. There's a molecule called CTLA-4 IgG1, which is a molecule that actually binds to co-stimulatory molecules on dendritic cells. Some of you will have heard of two molecules that are called CD80 and CD86. They're also sometimes called B7.1, B7.2. These bind to a molecule called CD28 on a T cell and provide a co-stimulation to activate T cells. CTLA-4 will actually get in the way because it will bind to CD80 and CD86. As well as CD28 binding to CD80 and CD86, CTLA-4 also binds to those two molecules. So it binds to CD80 and CD86, gets in the way so that CD28 can't recognize CD80 and CD86, and you don't get co-stimulation in the T cell. And then people also try and induce chimerism in the recipient, so that the recipient becomes tolerant. So it's almost like making a kind of an F1 mouse, where it's a hybrid of A and B. But here you have usually a microchimerism in the bone marrow that means that you're generating cells that are like the donor cells, and your immune system becomes tolerant. And this is just a slide to show the action of these three newer immunosuppressives that have really transformed transplantation: cyclosporine, tacrolimus, and rapamycin. Tacrolimus and cyclosporine, I'm not going to go in detail here, but basically they act downstream of T cell receptor signaling. So in order to activate a T cell, you have to signal through the T cell receptor here. And both of these drugs, tacrolimus and cyclosporine, block the signal very early on. In contrast, rapamycin acts later on. Because when T cells become activated, they start making interleukin-2. That interleukin-2 then binds to the interleukin-2 receptor on those T cells. And rapamycin blocks the signal from the interleukin-2 receptor. So tacrolimus and cyclosporine act very early on in T cell activation. Rapamycin later on, further downstream, once IL-2 is. So the future. Well, I mentioned, and we're going to manage to finish without having a break, so I'm just going to go on for another 15 minutes or so and then we'll be done, okay? So, the real problem now is not knowing what tissue typing to do, what MHC to match, or what immunosuppressive drugs to use. That has largely been solved. Still some debate going on, but in general, that's been solved. But there is this huge shortage of donors. Okay? So that's what people are really addressing now. And also, whether there's new types of transplant that can be carried out. I've mentioned the ones, heart, lung, kidney and so forth common, but are there other sorts of transplants that can be done? Well, there are, and in some places in the world, including Xi'an in China, people are doing face transplants, okay? Which is a very unusual thing. But if you have very severe damage to your face, you may want to have a new face. Now, this is something that psychologically is very difficult. We're all used to our face. Would we want a different face? Okay? So, there's a lot of caution in whether one should do this kind of transplant. People are doing this transplant. This was a transplant carried out at the 4th Military Hospital in Xi'an, and this is a paper published in Lancet last year, a 2-year follow-up. So 2 years after the face transplant, it was still surviving. Okay? And this is the picture. So this is his own face here, and this is a transplanted face here. So it's not the whole face, but about 50% of the face was transplanted. And this before was completely damaged. Okay? So it's given him a face that he can go out in public and feel that he doesn't need to look too awful. So you can transplant a face. What's the next step? If you can transplant a face, well, why not a brain? Okay? Now when you get to my age, your brain begins to sort of slow down, you forget things, okay? So maybe I'd like to have one of your brains, a younger, nice brain, so I could think a bit more actively. Might be very nice. Now it's very unlikely that people are going to transplant whole brains, but people already are actually transplanting nerve cells. So there are neurodegenerative diseases where the brain becomes damaged, and if you can transplant stem cells that will regenerate nervous tissue in diseases such as Parkinson's disease, then people are doing that, okay? So a whole brain transplant is science fiction, but transplanting nerve cells is not. You can take a blastocyst, so a fertilized egg that's developed into a blastocyst, you can isolate cells from this. I'm not going to go through the whole slide, but at the end of the day, from these stem cells, you can develop oligodendrocytes that will lead to remyelination of nerve cells. You can develop neurons that can restore circuits within the brain, and you can develop astrocytes that are cells that are involved in scaffolding and provide nutrition in the brain. So from stem cells, you can, depending on which particular molecules, growth factors and so forth you culture the stem cells with, you can drive them down particular differentiation pathways, and you can develop cells that you can transplant into the brain. People are doing this. This is something that people are beginning to do. So what other things can be done to overcome donor shortage? Well, one of the things that people have been discussing for many years is the idea of xenotransplantation. As I talked about at the beginning, maybe you could transplant tissues from one type of species to another. Okay? And really the species that people have investigated by far the most, pigs. Okay, so these are little baby pigs, quite cute. But there's a real problem with transplanting tissues from animals such as pigs in terms of immunological rejection. And this is a problem that I'll call the alpha-gal problem. We all have, whether we're humans or pigs or zebras or whatever, we all have lots of different proteins, and I've always mentioned those proteins can be modified. I said one of the kind of modifications that happens is glycosylation. And I was talking about cancer antigens, I talked about post-translational modifications. And one post-translational modification that can occur is glycosylation. And in glycosylation, you add sugars to proteins. And you can add many different sorts of sugars. And you can build up a sugar chain, an oligosaccharide, a structure made up of several sugars. And those sugars get linked in various ways. So when you put one sugar onto another, you can add it like this, or you can add it like that. You can add it in different positions. And the sugar has a 6-carbon ring, and you can add it to different carbons in the ring. And there is a particular linkage that can occur, which is a galactose, and then it has a particular linkage. It doesn't really matter, the details don't matter. It's called an alpha-1,3 linkage. You get one galactose sugar linked to another galactose sugar through what is called an alpha-1,3 linkage. Okay. Very late on in evolution, there was a mutation in the gene that makes that particular sugar linkage. So that certain higher primates, including humans, have a mutation in the gene that makes the alpha-1,3 gal linkage. So we do not have that particular sugar structure. We just don't ever have galactose, alpha-1,3 galactose, because we don't have the enzyme required to make it. But species below higher primates do have the enzyme in a fully functional form, and they do make that linkage. So pig cells have on their surface glycoproteins, and some of those glycoproteins have galactose, alpha-1,3 galactose. Now, unfortunately, if you go way, way back in evolution, down to bacteria, they can also make that sugar structure. And when we get infected with bacteria, because the bacteria are foreign, and because we don't have that particular sugar structure, alpha-1,3 gal, that's not a self-antigen to us, it's a foreign antigen. We're not tolerant to it, because we don't make that antigen, so we're not tolerant. So we make antibody to it. So we make antibody to alpha-1,3 gal. And amazingly, and this is quite incredible, several people have replicated this, about 1% of our IgG is capable of recognizing alpha-1,3 gal. So out of all the different structures that IgG can recognize, 1%, 1 in 100 of our IgG molecules can recognize this structure. So we have masses of antibody against alpha-1,3 gal, because it's very common on bacteria. The problem is, it's very common on pig cells as well. So if you take a pig, then you'll get immediate hyperacute rejection. Remember when I talked about different types of rejection, the fastest is hyperacute, almost immediate, within minutes, because there's pre-existing antibody. And we all have lots of antibody against alpha-1,3 gal. So if you transplant a pig tissue or organ, then the natural anti-gal antibody that we have will bind to the organ, complement becomes activated, and the organ is rejected immediately. So that has been the major hurdle to using pigs as donors for tissue for humans. The other issue, just I might as well mention, is that people are also concerned whether there may be viruses in pigs that would be transferred to humans and cause disease in humans. What are called zoonoses, which are viruses that you normally, or other infectious agents you find in one species that can potentially infect another species. So there is concern about that. But the immediate problem in immunological terms is this immediate rejection. So how can you get around that? Well, one idea is that in the pigs, you make a knockout pig. Just like you can knock out genes in mice, you can knock out genes in pigs. You make a knockout pig where you've knocked out this enzyme, so it can't make that structure. So effectively, our gene has been knocked out by mutation. So we don't have functional alpha-1,3 gal galactose translation. So maybe you can make a knockout pig. Okay? Then it wouldn't have any alpha-1,3 gal, wouldn't be rejected. Or another approach that people are using, and there are companies, commercial companies that have done this in the hope that they'll be able to sell the organs for transplantation. You can make transgenic pigs where you put in a molecule called DAF, which stands for decay accelerated factor, or another molecule CD59, one of the CD antigens. And both DAF and CD59 are what are called complement regulatory proteins that prevent complement killing our own cells. Because when we get an infection and an antibody binds and it activates complement, that complement could potentially damage our own cells. So we need molecules that inhibit complement being activated on our own cells. And these two molecules, DAP and CD59, are examples of those molecules. So maybe you can make the fix transgenic for human DAP or CD59, and they would be protected against complement-mediated damage. Or, yet another approach that people have used is to transfer other kinds of transferases into the cells, so that you then put other sugars on the end. Because what is recognized by the natural antibodies we have is gal alpha-1,3 gal at the end of a sugar chain, what's called a terminal pair of oligosaccharides. Then you could cover that up with other sugars and it wouldn't be recognized anymore. So if you make pigs that are transgenic for alpha-1,2 fucosyl transferase, then you have gal alpha-1,3 gal, but you cover it with a fucose sugar, and that can no longer be recognized by our natural antibodies. So these are all things that people are doing to try and overcome this problem of us having 1% of our IgG against the structure that is present on the surface of pig cells, the carbohydrate structure. And then another thing that people are doing is trying to make artificial organs. So there's not enough donors. Maybe they'll be able to use xenografts from pigs or other animals in the future, but the technology is still being developed for that. So can you make artificial organs and tissue? So people are using what is called tissue engineering. So maybe you can take cells, you can take stem cells, you can grow them on a matrix that would mimic a particular structure, and then you could grow that in the lab, and then you could transplant that back. So maybe you could produce an artificial liver where you've taken stem cells grown under certain conditions so that they're differentiated into hepatocytes and other types of liver cells, grow that. So they are made. But generally speaking, they're not very reliable. After a while, the immune system might, or other body systems might start to sort of, think, I don't really want this thing inside me. And, you can get sort of damage and to surrounding tissue because of inflammatory responses that are going on and so forth. So they're good as a sort of stopgap measure. If you're waiting for a transplant and you haven't quite got one, sometimes they put these things into people while they're waiting for a transplant. But they don't seem very reliable at the moment, long term. But they may be in the future. And then I just want to finish just the last two slides, or the last slide really, and then a summary, is that there is a situation of a transplant that occurs naturally. Transplantation is a very unusual, very unnatural thing. Only been going on for 50 years as regards things that we think about as transplants. But something has been going on since there's been human beings that is also a transplant, okay? Which is the fetus. When a woman becomes pregnant, half of the antigens in the fetus are from the father. Half are from the mother, but half are from the father. Okay? So those paternal antigens from the father in the fetus can potentially be recognized as foreign. And it's long been puzzled by immunologists for several decades now. Why doesn't the mother reject what, after all, is a semi-allogeneic transplant? Okay? Half of the genes are from the father. And we still don't know why. Okay? There are many things that are proposed, and this slide just gives some idea. And one of the things I will point out is I mentioned that we have three major sorts of MHC class 1 molecules. HLA A, B, and C. But we have other ones. HLA G, E, and F. Those are other MHC class 1. But they are not expressed on most cells in the body. But they are expressed in the placenta. And it's thought that maybe they're expressed in the placenta to stop natural killer cells actually attacking the placenta. Because what there isn't in the placenta is what we can call the classical MHC class 1 and class 2 genes. So there's no HLA A, B, and C. And what natural killer cells look for is an absence of class 1. Natural killer cells kill cells that are not expressing class 1. Normally, all cells express MHC class 1. But before the break, I said tumour cells sometimes don't express MHC class 1, and natural killer cells can kill them. But in the placenta, if you don't have class 1, then natural killer cells would kill the placenta. But maybe the presence of these other types of class 1 molecules, G, E, and F, forget that. There are also complement control proteins that are expressed. There's also a class ligand, that if any cells of the immune system did make their way to the placenta, maybe FAS would be engaged on the cells of the immune system and would induce apoptosis in the cells of the immune system. There's also the fact that the placenta acts as a kind of physical barrier. There's also lots of immunosuppressive molecules that are produced in the placenta, such as transforming factor beta and interleukin 10 and an enzyme called IDO. All of those are immunosuppressive. And it seems that maybe in some parts of the placenta there are actually some molecules that can act to absorb out any antibodies that would be against MHC. So a whole host of different mechanisms have been proposed. Which of those are the most important, or whether there are other mechanisms that are still not understood, is not known yet. But it's a very interesting transplant situation, the fetus as an allogeneic transplant. So, in summary, when you transplant a tissue or organ, you get immunologically mediated rejection. Successful transplantation requires at least some degree of tissue typing, but it may not be necessary to completely match every antigen, but some degree of tissue matching is needed. And you also always, unless it's an identical twin, you need immunosuppression. And the real problem now is a lack of donors. Okay, that's it. Any questions related to transplantation? Okay, well, as I say, afterwards, if any of you are too shy to ask questions in front of the group, you want to come up and ask me questions afterwards, I'm more than happy to do so. It means the group don't benefit from the answer, which is a good reason to ask them when the whole group is here. But if anybody does want to come up after and chat to me, that's absolutely.
Do you have an email address?
Yes, I do have an email address, and it's quite simple. I haven't got a pen, I can't write it down, but I'll tell you, okay? So this is in lowercase letters, P dot delves, which is DELVES. Okay, I'll put it onto a slide for later in the week. It's P dot delves at, so P dot delves at UCL. Stands for University College London, UCL, .ac, stands for academic, .uk, United Kingdom. Okay. So all in lowercase letters, p.delves at ucl.ac.uk. But I'll put it on a slide for later in the week. Yeah, please. Quiet!
Technical one is clinical speech. As we all know, that if after transplantation, we should use drugs of immunosuppression to prevent rejection from the recipient. My question is does the drug function specifically within the area of a transplanted organ, or does it have effect to the whole immune system of our body? And if our immune system is suppressed, does it mean that we are more vulnerable to injections from the outside and maybe cancer?
Yeah. Okay, so the question is, when you give immunosuppressive drugs, do they just act at the site of the transplanted organ or tissue, or are they acting throughout the body? And if they're acting throughout the body, because we've got immunosuppression, do we get more cancers, do we get more infections, okay? So the answer is that they act throughout the body, and it would be very good to develop drugs that acted specifically at the area of the transplant. So that is something that certainly people could be trying to do, is develop immunosuppression that is just local to the transplant rather than throughout the whole body. But the immunosuppression is systemic; it happens throughout the whole body. As I mentioned in the first half of this afternoon, if you look at people who are getting transplants, you do see higher levels of lymphomas, for example, not of most types of cancers, but certain types of cancers, particularly lymphoma that's associated with Epstein-Barr virus. So it seems that there is an effect on combating infection with Epstein-Barr virus that then allows it to go on and cause lymphoma in patients that are immunosuppressed because of transplantation. In terms of other types of infections, you do see an increased level of other types of infections. So that is something that the clinician needs to be aware that the patient may get other types of infections. But very interestingly, when you do suppress immune responses, often you don't get the very high level of very diverse infections that you might predict you would. Because a lot of suppressive mechanisms, somehow it seems that other parts of the immune system can compensate and still fight the infection. So maybe, for example, you might be very much dampening down a cytotoxic T-cell response, but maybe, the innate response with macrophages and neutrophils and eosinophils and perhaps other parts of the adaptive response like antibodies can compensate. So it really depends how broad the immunosuppression is. And usually there'll be some increase in infection, but it's not huge and it's manageable. You just very proactively treat with antibiotics. Okay, that's a good question. Anybody else? Okay. Right, thank you very much for your attention.